Hello guys, in today's lecture we will cover CVS infections of microbiology. We will cover infections of heart, infections of blood vessels, device-related infections and autoimmune-mediated infections. Infections of heart, there can be involvement of three layers: endocardium, myocardium and pericardium. The infections are endocarditis, myocarditis and pericarditis. Device-related infections, we will learn about catheter-related bloodstream infections, suppurative thrombophlebitis. In autoimmune-mediated infections, there is acute rheumatic fever, and in infections of blood vessels, there is mycotic aneurysm and infective endocarditis. Now we will learn infective endocarditis. It refers to the microbial invasion of the heart valves and mural endocardium. It results in formation of bulky friable mass, which is composed of platelets and fibrin, microcolonies of organisms, and scanty inflammatory cells. Most, the most common site of vegetation formation is heart valves, low pressure site of ventricular septal decay and mural endocardium. Now the classification of endocarditis, it can be divided into acute endocarditis and subacute endocarditis. In acute endocarditis, it shows rapid evolution. Subacute endocarditis shows slow evolution. Acute endocarditis mainly involve normal cardiac valves. Subacute endocarditis mainly involves previously damaged heart valves. So in acute endocarditis, there should be implicated organism should be highly virulent to cause damage to the normal cardiac valves. So the involved organism is Staphylococcus aureus. The subacute endocarditis is mainly occurs in the damaged heart valve. So the organism is of low virulent organism. In the morbidity and mortality even after appropriate antimicrobial therapy is much higher in case of acute endocarditis, whereas in subacute endocarditis it easily recovers after antimicrobial therapy. Acute endocarditis is less common, only 10 to 20% cases happens, whereas subacute endocarditis is most common and it is 50 to 60% of cases. The etiology, that is the causative agents of the infective endocarditis, they are mainly Staphylococcus aureus, coagulation negative Staph aureus of breed and streptococci, enterococcus and pneumococci and mainly important group has a group. The full form is very important. It includes Haemophilus influenzae, Campylobacterium hominis, Candida colorens, and Klebsiella kingi. Now the pathogenesis of infective endocarditis, the risk factors includes underlying cardiac defects, that is mitral regurgitation, or the IV catheters and prosthetic valve surgery previously done. This predisposing factors damages the endothelial surface of the heart, which causes deposition of platelets and fibrins in the endothelium, which causes thrombus formation. Thrombus formation leads to a condition known as NBTE or non-bacterial thrombotic endocarditis. Mainly transient bacteremia after brushing teeth or taking food, chewing food. There is transiently bacteria gets access to the blood supply and the bacteria adhere to the thrombus, which leads to colonization of bacteria. When the vegetation formation occurs, the deposition of platelets, fibrins, and inflammatory cells surrounding the organism, which produces vegetations. Basic vegetations seed bacteria into the blood at a slow but constant rate, and they metastasize to distant sites. And it is the pathogenesis of infective endocarditis. Now, the diagnosis of infective endocarditis, it is mainly dependent on modified Gibbs criteria. Or confirm the diagnosis, there should be fulfillment of the two major criteria, or one major plus three minor criteria, or five minor criteria. Learn the major criteria. First of all, positive blood culture. If there is presence of any one of the positive blood culture, then we will confirm the case. It includes typical infective endocarditis organism isolated from two separate blood culture sets. Blood culture sets means blood collected from single manufacturing site and divided into two bottles. It is called positive if any of the two bottles is positive. Then secondly, persistently positive blood culture with agents other than typical infective endocarditis organisms. And thirdly, single positive blood culture sets for coaxial variety. And secondly, the major criteria is evidence of endocardial involvement in echocardiography or there is new valvular regurgitation. In echocardiography, it is mainly showing oscillating intercardiac mass on the valves or abscess or new partial dehiscence of the prosthetic valves. Now the minor criteria are there can be predisposing factors like heart conditions or IV drug uses. It's a second with a fever greater than 38 degrees centigrade. With muscular phenomenon like arterial emboli or pulmonary infarcts. Mycotic aneurysm, mainly important part is GNMA lesions. Immunological phenomena like important is ocular snots, rock spots, and rheumatoid factor. Microbiological evidence like positive blood culture but not fulfilling the major criteria. The serological evidence are present. Thanks for watching. If you like, share with your friends and subscribe to MEDICO.